Okay, so welcome back, friends. From today, I will start another video lecture series that where I will focus on viral transfection. This is another kind of gene delivery method that we have to discuss. So, before going to enter in the introductory video of viral transfection, I would like to recommend you to see all the gene delivery system videos that I have uploaded earlier. In case if you do not have any kind of knowledge regarding other gene delivery system, okay, because viral transfection is the most modified one. So you have to learn about the basic transfection or transformation procedure first, then it will be easy to you to understand about viral transfection. And if you have knowledge about other kind of gene transfection method, then let's start to learn about viral transfection method. So far, before going to enter in detail, I want to mention you that the term transfection is nothing new. Okay, so this transfection simply is a name of transformation. Okay, that means when you are transferring your gene of interest with the help of a virus, then instead of transformation we are using transfection term, or more specifically the term transduction. Okay, so let's discuss about the viral transfection. So the idea is viral transfection is, or viral transfer, or viral mediated gene deliver system is employed to transfer, or to transform, I should say, those cell type that cannot be transfected with the help of other gene delivery system that we have. Okay, so viral transfection method provide us the opportunity to transfect those cells that cannot be transfected with the other type of gene delivery system. As a result, now we can express or overexpress our protein of interest in those host cell types too. Okay, so, and it is the most commonly used method in clinical research. Okay, so in that slide we have learned about the viral transfection definition, that is a type of gene delivery system mediated by virus that helps you to transfect those cell type that cannot be transfected with the help of other gene delivery system that we have. Okay, so expression of protein in those cell types nowadays possible because of the viral transfection, and it is mostly used in case of clinical research purposes. Okay, now we have various kind of viral vector system available. So among them, the most important types are adenoviral, oncoretroviral, and lentiviral vector. Okay, or along with baculoviral vector and vaccinia virus vector are also important. Although we have discussed about baculoviral vector earlier, right? It is used to transfect mainly insect cell, right? So if you want to know about baculovirus, then I will again like to recommend you the video that I have uploaded regarding baculovirus. Then you can understand about what is baculovirus based vector. Okay, so we will discuss about each of the vectors gene delivery system one by one in our upcoming videos. Okay, so now we are going to discuss about what are the advantages and what are the disadvantages of viral vector. So in your left side, these 2 points are the advantage of viral vector. One is the high transfection efficiency in vivo. Okay, it is highly efficient transfection procedure, because remember, virus can only transfect those cells that have specific receptor for that virus. Okay, as a result, you don't need to bother about non-specificity that we are facing in previous gene delivery systems, right? So viral vector provide you a high specificity. Okay, along with it will also allow a sustained gene expression. Why? Because it will help you to integrate your gene of interest with the host genome. Okay, for example, in case of baculoviral vector, we have seen that baculoviral vector integrates itself with the host genome, right? So that is the idea. As a result, it will facilitate sustained gene expression that you cannot get in case of when you are using plasmid as a vector and E. coli as a host, right? It will not integrate inside the bacterial genome, right? It will remain as a plasmid. So when another conjugation procedure happens by chancely with another plasmid, another bacteria, then your gene of interest may be transferred to that one. That can be a problem, but viral vector ensures those kind of situation, because here the sustained gene expression due to integration. Okay, that is the two important advantage that viral vector provides. Again, the high in vivo transfection efficiency, and the second one is the sustained gene expression, as viral vector or viral delivery system allows integration of gene into the host genome. Okay, now let's discuss about the disadvantage parts. So it have immunogenicity, obviously that is nothing new, cytotoxicity, technically challenging, and there is a laborious amount of production procedure you have to do for production of the vector, along with high cost due to the biosafety requirements. Okay, because remember, here we are dealing with oncoretroviral, that means it is cancerous, so we have to develop a good biosafety requirement, and that will be obviously costly, and variability in the infectivity of the viral vector preparation. Okay, that is another kind of disadvantages, because there is a variation in the infectivity of the viral vector preparation. So different type of virus infects differently, so you have to keep in your mind during using them as a gene delivery system, along with the low packaging capacity. Okay, so viral vector provides you high in vivo transfection efficiency along with a sustained gene expression, as it helps our gene of interest to integrate into the whole genome. Along with that, it also provide a couple of disadvantages. For example, a good technical skill required, and it is also technically challenging procedure. It have the ability to provide immunogenicity, cytotoxicity, along with laborious amount of work is required for the production of those kind of viral vector systems, along with high cost due to the requirement of the biosafety measurement, biosafety system, along with low packaging capacity. Okay, so now let's move to the next slide, where we are going to discuss about a skeleton of a standard transfection procedure. Okay, although we are going to discuss about each transfection procedure for each viruses, adenoviral viruses, lentiviral viruses, along with our oncoretroviral, each of them. Okay, so the first thing is we will engineer the virus. That means here the virus is recombinant virus containing recombinant viral particle carrying the transgene. Transgene means is nothing new, it is our gene of interest. Okay, after the construction of the recombinant virus, we will transfect them. This, now mind it very carefully, what I want to mention, that here the transfection procedure is non-viral. Okay, and here we are transfecting it in a packaging cell line. Okay, so in the packaging cell line it will amplify. As a result, amplified recombinant viral particle will obtained. So then we will purify them and we will titrate them with the help of titration. After that we will apply them in our cell of interest. Okay, so there are two-step processes, right? First of all, we will engineer our recombinant virus, and now our virus contain the transgene, that is our gene of interest. Then we will transfect them in a packaging cell line for amplification. After amplification, the amplified viral particles will be purified and we will also apply the titration on them. Then we will apply subsequently them into another cell of interest. Okay, that is our main transfection procedure. Here one thing I want to mention, that most of the primary cell and cell line show a 90 to 100% transfection efficiency when you are using a viral vector. Okay, but here there is a problem, that is the specificity. That means if your host cell do not contain the specific receptor, the transfection procedure will not going to happen. That means as well as it will provide you a good specificity, no doubt about this, but it is as well as a disadvantage too. That means you cannot use any kind of cell for as a host when you are using viral vectors, right? You have to keep in your mind that what kind of cell I can choose that will have a particular receptor that allow me to enter my virus containing gene of interest inside it. Okay, so that is the idea of viral transfection. So in the first video of viral transfection, you have learned that viral transfection is another kind of gene delivery system where you are transferring your gene of interest into a suitable host with the help of virus. Okay, and it is mainly used for transfecting those cell type that cannot be transfected with the help of other cell type. Okay, and remember one thing I want to add another point here, that as here we are using virus, so it will obviously be harmful. Okay, so it will only be used for those cell types that cannot be transfected with the help of other methods. Why I am saying this? Because you may get some MCQ question where they will ask you that what will be your preferable method for gene delivery. Okay, they will give you a story and then they will ask you that we are willing to insert a gene of interest. Now you tell us that what kind of procedure will we can choose. Then I would like to recommend you to read the question very carefully. If they are telling that transfection is very difficult, then go for viral transfection, otherwise not, because it is harmful. That is why researchers do not willing to recommend this kind of transfection when the transfection can easily be done with the help of other method. Okay, that is another point. So that is the idea of viral transfection, and there are three main type of viral system that we are using. One is adenoviral vector, another one is oncoretroviral vector, another one is obviously the lentiviral vector system. Okay, so then we have discussed about the advantage part, that it will give you a high transfection efficiency in vivo and the sustained gene expression due to the integration of the host genome by the viral transfection into the host genome. Okay, so here it will be the integration of the transgene, right, not host genome, in the host genome. Okay, so then we have discussed about the disadvantages part, that is it have immunogenicity, it have cytotoxicity, it have a laborious amount of work needed to prepare the viral vector. Along with it require cost, because the development of a good biosafety system, right? And also the packaging, it provide only a low packaging capacity. So here the typical transduction procedure: first of all, the virus will be genetically engineered carrying the transgene, then you will transfer them in a packaging cell line where it will amplify. After that you will isolate them again. Okay, so after isolation you will purify them and titrate them, then you will transfect them in your desired cell types. Okay, so that is how the entire procedure of typical transduction protocol in case of virus going on. Okay, and another important thing, yes, it will provide you 90 to 100% of the transfection efficiency, but you have to make sure that a specific receptor present on the surface of the cell. Otherwise the viral transfection efficiency for those kind of cell will be 0. Okay, so that was the first video regarding viral transfection. So I hope this video will be helpful to you, and thank you for listening to this class. We will again come back in another video where we will going to discuss about the key features of the viral transfection. Okay, so thank you, take care.